Okay, so the tertiary structure of proteins is basically a complex arrangement of the secondary structures, and it exists in the form of domains. So we should be knowing that what is domain. Domain is an independent fundamental functional and structural unit. It is an independent and structural and functional unit of protein. So it is structural unit as well as functional unit. It means that its folding is independent of other domains. So folding in one domain is independent of folding in the other domain. How it occurs? For example, this was the primary structure of protein. It arranged into the different secondary structure. This primary protein structure arranged into the different secondary structures and formed multiple alpha helix, uh, beta strands, alpha helix, beta strands, alpha helix, and so on. And these alpha helix and beta strands arrange themselves into the domains. For example, this beta strand and alpha helix form one domain. So alpha helix, beta strand, alpha helix, it formed one domain. Then there existed a coil between them, and then here alpha helix, beta strand, alpha helix, beta strand, and so on. So it formed a other domain. This is, we can call it one domain, and we can call it second domain. So these both domains will undergo folding independent of each other. They both undergo their folding, and then these two domains, then they undergo complex folding. These two domains then will fold upon itself. So folding in domains individually independent of each other, and then these domains combine and give a complex structure to the protein. So this is the tertiary structure of protein. And how it is stabilized? This tertiary structure of protein is stabilized by disulfide bonds. Disulfide bonds, it is stabilized by hydrophobic interactions. It is stabilized by hydrophobic interaction and hydrogen bonds, and it is stabilized by ionic interactions. So ionic interaction. So what is disulfide bond? We previously discussed that cysteine and cysteine residue. Cysteine are two sulfur containing amino acids. When they come close to each other, they interact and form a disulfide bond. So supposedly in amino acid chain, here was a cysteine residue, then chain continued and here was a cysteine residue. When chain folded upon itself, then two cysteine residue came close to each other. When chain folded, so as a result of folding, one cysteine residue came in front of other cysteine residue. So these cysteine residue, when they came in front of each other, what they happened, they interacted and formed disulfide bonds. And these disulfide bonds give stability to the tertiary structure. Similarly, hydrophobic interaction occurs. Protein has both hydrophilic and hydrophobic amino acids. So supposedly this is the chain here. When it is subjected to water or solution form, what happen? The hydrophobic amino acid will get buried inside, while hydrophilic amino acid will come on the surface. So hydrophobic interaction imparts stability to the tertiary structure of protein. Next, hydrogen bonds formed due to polarity of oxygen and hydrogen due to amino and carboxylic group. And then comes ionic interaction, which are basically due to charged chains as in acidic and basic amino acids, aspartate, glutamate, histidine, lysine, which have charged R chains. Charged R chains, which can lead to the formation of ionic interaction. So disulfide bond, hydrophobic interaction, hydrogen bond, and ionic interaction impart the stability to the tertiary structure. So this linkages are basically asked in question as describe the role of R group. Describe the role of R group in the structure of protein, determining structure of protein. So what is the role of R group? R group lead to the formation of disulfide. R group can lead to the formation of hydrophobic interactions, and moreover it can lead to the formation of ionic interaction. Hydrophobic interaction and ionic interaction are principally due to R group of the amino acids. Okay, so it is protein folding. We can assume it here betterly from the diagram. This is the, if you see here, this is the primary polypeptide chain. Then it is assumed the secondary structure. Here is our beta strands and alpha helix. Then these beta strand and alpha helix arrange themselves in the motifs or domains. These are the domains, combination of alpha and beta helix. Then these domains arrange into a complex protein structure, which is the tertiary three-dimensional structure of proteins. Okay. So next we move on about the denaturation of protein. What happens in denaturation of protein? In denaturation of protein, protein's primary structure is maintained. However, the higher structure, secondary, tertiary structure of protein is lost because there is breakage of the hydrogen bonding and so on. So secondary and tertiary structure of protein is lost in denaturation. What is denaturation? Denaturation is the loss of secondary and tertiary structure of proteins. Okay, so denaturation can occur by mechanical condition as well as acid as well as heat and so on. Next we move on toward discussing the role of chaperones. Role of chaperones in the determining the structure of proteins. So what are chaperones? Chaperones basically are specific proteins. These are specific proteins which regulate the protein folding. These are specific proteins which regulate the protein folding. These are also called heat shock protein. These are also called heat shock protein. So what they do, initially it was supposed that the primary structure of protein is responsible for folding. However, in denaturation the primary structure is maintained by a denatured protein does not retain its secondary and tertiary structure. So it may, if it was then thought that there must be something else. So chaperones are basically proteins which regulate the protein folding, and they can keep protein folding starts during the translation when even whole polypeptide chain is not formed, and it prevents the protein from getting into unnecessary interactions. And these chaperones, they can keep the specific areas protein unfolded, or they can lead to the or accelerate the specific areas of protein into the folding process and so on. So in this way the chaperone regulate the protein folding. Next we move on towards the quaternary structure of protein. Next we move on towards the quaternary structure of proteins. So what is quaternary structure? Quaternary structure. The tertiary structure of protein is assumed by one polypeptide chain. A protein having one polypeptide chain can go up to the tertiary structure. Tertiary structure. However, protein having multiple polypeptide chain, if protein has multiple chains, polypeptide chains, then this protein attains quaternary structure. Quaternary structure. For example, myoglobin attains tertiary structure, while quaternary structure is attained by hemoglobin. Rest all things are same. Quaternary structure also have similar the alpha helix, beta pleated sheet, and then their domains, and then their complex folding. Similar tertiary structure also have similar foldings. However, the quaternary structure, due to presence of multiple chain, it is more complex. It is more complex, and it involves more than one polypeptide chain, and it is less complex. And the interaction, they are stabilized by, both are stabilized by same interaction. For example, hydrogen bond, hydrophobic interactions, ionic interaction, and disulfide bonds. So it was about the tertiary and quaternary structure of proteins. Thank you.